go on. So hello and welcome to all of you to the Science Setu lecture series 4 by Dr. SREEDHAR Chinnaswamy. So before starting our Science Setu lecture 4, I would like to request our principal sir to please share his words of wisdom. Sir, please.
So good afternoon to you all. Good noon, better to say. Thank you, Nivedita ma'am, and thank you, sir. So first of all, I would like to express my sincere gratitude and heartfelt thanks to Dr. Chinnaswamy, SREEDHAR Chinnaswamy, Associate Professor, NIBMG, for giving his consent, and he'll express his valuable experience and vast research experience. And students, we are very lucky enough, I think. And not only the students, I have seen many of my colleagues, the departmental coordinators of DBT Star College scheme, they have also joined. Thank you all the departmental coordinators and obviously Nivedita ma'am, coordinator of DBT Star College scheme. Students, we are very lucky enough, I think. And one my honest request to the students, so this is the Science Setu lecture series, and today is the 4th as mentioned by ma'am. So please interact with such an eminent resource person after completion of the session. So if you have any question, you raise the questions, and sir is ready to answer your all the queries. Not only the students, faculty members, you are also requested to interact with sir. So with these few words, once again, my heartfelt thanks and sincere gratitude to sir. So I wish you all the grand success of the seminar, and my warm welcome to you, sir, in the physical mode in your college when the situation will be normal. Thank you so much.
Thank you.
Thank you, sir, for your kind words. So before starting the series, I would like to introduce our resource person to our students and faculty members. Dr. SREEDHAR Chinnaswamy, Associate Professor at the National Institute of Biomedical Genomics, completed his bachelor's degree from Veterinary College, Bangalore in 1999 and his master's degree from Indian Veterinary Research Institute, Uttar Pradesh. He then completed his MS from New Mexico State University, USA, and was awarded PhD in 2010 from Texas A&M University, USA. He has several awards and honors to his credit, which include Dean's List, New Mexico State University, USA in 2005, member of the Infectious Disease Biology Task Force, Department of Biotechnology, Government of India from 2014 to 2018, the Visiting Fellow of the Jawaharlal Nehru Center for Advanced Scientific Research 2012 and Visiting Scientist of the Healthy Aging Research Center, Department of Immunology, Rheumatology and Allergy, Medical University of Lodz, Poland 2015 and Intermediate Fellow of DBT Welcome India Alliance 2017. His research interests revolve around the genetics and biology of type 3 interferons. Type 3 IFNs, also known as IFN Lambda, are a recently discovered family of IFNs that are thought to be the critical players at the host-environment interface involving the epithelial services. With his vast research experience, he has published several research articles in very reputed high-impact factor international journals, guided doctoral students, completed major research projects funded by Department of Science and Technology, the CRB, and Department of Biotechnology, Government of India. He has delivered several international invited and special lectures as well. Sir, we are very happy to have you here. Sir will be speaking on genetics and immunobiology of interferon lambda 3/4 and their significance in human health and disease. So I would now like to request Sir to please deliver the technical session. Sir, please.
Thank you very much, Dr. Nivedita, and also Dr. Devnath, who spoke before, for this kind introduction about me. And I'm really, happy and glad to be participating in this lecture series, which is very, a very important initiative taken by you, all the organizers, and also I think DBT InSTEM is involved in this. So it is always an important aspect in an academician's career to interact and motivate youngsters to come into science and feel and have a feel for what science is and get excited by science. So this is a very good forum, I think, where most of them will be youngsters, as I understand, who will be watching this lecture. So I'm really glad and happy to be doing this for you. So without further ado, let me start my presentation. Let me see. Okay. Yeah. Are you able to see my screen now?
Yes, sir. Your screen is visible.
Okay. I've just gone to the full view mode. Are you able to see that?
Yes, sir. I am also able to see that.
Okay. Is the slide moving?
Yes, sir. Slide is also.
Okay. Fantastic. Thank you. Okay. So again, thanks, Dr. Nivedita, for briefly introducing my area of research. So I have been interested in this area of research, which is genetics and biology of type 3 interferons, which are also called as interferon lambdas or lambda interferons, whichever way you want to call. Since I have been to this institute called National Institute of Biomedical Genomics, now it is called as BRIC, NIBMG. BRIC stands for Biotechnology Research Innovation Council, NIBMG. So this is a recent merger of institutes that has happened from the DBT side. So we are, from now on, we will be part of the BRIC umbrella of institutes. So we are BRIC NIBMG. So I joined this institute in the 2010 when I came back from the US and started my research career as an independent researcher in this field. So, and I will be happy to share my results in this next half an hour, 45 minutes or so about what I have been doing in the last 4 to 5 years. So I know this involves a lot of youngsters who may find it a little difficult if the talk is too technical. So I've tried to, I will be trying to be very as simple as possible and be as fundamental as possible in introducing concepts, etc. But if there are any problems, any doubts, you can always put it in the chat box and I will get back to you at the end of the lecture. Okay, so now let's start from the very very basics of I told you I work on type 3 interferons. Let me make my this thing here. How do I get my okay pointer option? Actually, okay, yeah. Are you able to see my pointer now?
Yes, sir. The
okay, great. Okay, so as I told you, I work on this class of interferons called as type 3 interferons. But along with type 3, as you see, there are type 1 and type 2 also, which are the 3 classes of interferons you generally get to see in any mammalian biology. So as you see, type 1 interferons, a lot of them are there, and these are the well-studied ones because they have been studied from a long time. Mainly the interferon alpha, which has several of its isoforms, and about 4 or 5 other kinds of type 1 interferons. Now type 2 interferons has only one member, interferon gamma. Type 3 interferons, the ones that I'm interested in, basically comprise of 4 interferons as named here, IFNL or interferon lambda 1, 2, 3 and 4. So these were only discovered in 2003. This 1, 2 and 3 was discovered in 2003 after the Human Genome Project. Interferon lambda 4 is even the newest member of this family, which was only discovered in 2013. That is about 11 years back. So I have been working in this new interferon under my Welcome DBT project, which I just concluded. So anyways, so the type 3 interferons are very important because they are called as the frontline guardians of the immunity and homeostasis in the respiratory tract, or in a more broader sense, they are called as the guardians of the epithelium. Now what is epithelium? Epithelium consists of the largest tissue, so to say. For example, your respiratory epithelium, your reproductive tract epithelium, your digestive tract epithelium. Not only that, your skin is also made up of stratified squamous epithelial cells. That means a large tissue of the human body consists of this epithelial cells, and these interferons are now considered the guardians of epithelium. In other words, the epithelium is the doorway to the outside world. So you are defined in this world or universe by the interaction that you have with the outside world through your epithelium. So to say, biologically you are connected to the outside world through your epithelium. So therefore, these kind of interferons which work in this epithelium specifically become very important players to study. And for example, in this figure, it is shown that the first line of defense against a viral infection in the respiratory tract is the type 3 interferon. They are very potent, antiviral factors, and also they are less harmful to the host. So for some reason, if the type 3 interferons fail in clearing the virus infection, then kicks in the type 1 interferons, which are more robust, of course, which can more efficiently get rid of the virus infection, but the trade-off will be that there will be some amount of tissue damage. So it is better that your infection stops at the type 3 interferon level. So that is the importance of type 3 interferons. Now, I told you there are 4 interferon lambda genes. So there is this figure or schematic shows you in a more better way. So as you see, 1, 2, 3, and 4 are 4 interferon lambda genes present on chromosome 19 of humans. Now, these boxes basically refer to exons, and in between 2 exons, the line you see is an intron, and between 2 genes, it is the intergenic region. So if you see that around 50 kb region is this locus called the interferon lambda locus. Now you see a lot of numbers around here. So what is this numbers mean or stand for? These numbers actually are representing the variations present in the human population. For example, this refers to if you might have heard of the term single nucleotide polymorphism. So these are basically referring to single nucleotide polymorphisms in this locus. For example, when I say RS4803217, RS stands for reference SNP ID number 4803217. I will try to take only the last 3 numbers of any SNP ID from now on. So for example, RS217, you might have a C nucleotide here, or you might have a T nucleotide, depending on which population you belong to, depending on your ethnicity, etc. But this is the variation that has been recorded in the human population. So as you see, there are a number of such variants spread out across this locus. Now, important amongst them are boxed here because these 3 variants rather are known to affect the function of the genes they are associated with. For example, RS813, which is present in the promoter of this gene called interferon lambda 3, and RS217, which is present towards the end of this gene called interferon lambda 3. Both of them, the alleles affect the expression of this gene. For example, if you have one kind of an allele, you might see one kind of expression level of lambda 3, and if you have the other allele, you will have a different level of expression. Since these are antiviral molecules, it becomes the level of expression becomes important in a virus infection. For example, if you have the better allele, you will have more interferon lambda, which is good for you to get rid of the virus infection. So that is the meaning. Another important variant here is RS815. This actually is not a single nucleotide polymorphism, it is a dinucleotide polymorphism. The reason is there are 2 nucleotides in this polymorphism that are changed. For example, you may have the TT at this position, or you may have delta G. Means one T is removed, that is delta, the other T is replaced by a G. So you know the ATGC. I'm sure everyone of you who have taken biology at some level in your schools, colleges, you know what nucleotides are, the 4 nucleotides. So I'm talking about the T nucleotide or the G nucleotide here. So the interesting thing in this dinucleotide polymorphism is that only people who have the delta G variant of the allele, we could also call the gene as an allele. So variant of a gene is called an allele, right? So if you have the delta G allele, only then you, as you see, this gene can produce number of isoforms. So you might heard of transcription, right? So from the DNA, your DNA becomes RNA through transcription, and then RNA becomes protein through translation. So when this gene is transcribed, interestingly, you see number of isoforms. We call it different. If the gene product is one, one DNA becomes one RNA, one kind of form, that is one gene getting transcribed. But here in this case, a number of different forms are coming. So if you see the TT allele, you have 5. If you have delta G allele, also you have 5. But only if you have the delta G allele, you get an active interferon lambda 4. That means the new interferon lambda 4 comes only in those people who have the delta G allele. So, for example, if you belong to the delta G allele group, then actually you have an extra interferon lambda, which is interferon lambda 4, compared to the people who don't have that allele, they instead have the TT allele. So this is the importance of this polymorphism. So depending on which allele you have, you might have an extra interferon lambda or not. And therefore it becomes important to study the group of people who have this extra interferon lambda 4 compared to the ones who don't have, and see their response to diseases as well as in health also, how they are different. So that is my interest here in my projects here. This is the basic interest revolving around different projects that I pursue here. Now this locus, this entire 50 kb region, 50 kilobases region of the genome is also under strong selection. That means as humans migrated out of Africa and settled in different regions of the world, this frequency of these polymorphisms keep changing. That means the way we have adapted to our life or lifestyles after we have emerged from Africa has some alleles, because they keep changing in different geographical regions and time points during human evolution. So that means it is evolutionarily important locus. So that is why I am interested to study in the Indian population specifically how these different polymorphisms here, they are playing a role in defining the biology of interferon lambdas and how their importance is there in diseases and also in health. So I will try to explain some results that I have obtained in the last 4 or 5 years here. Now this locus I told you was initially discovered in 2003. But until 2009, this was, it was kind of only for academic importance, because there was a type 1 interferon which was very well studied, so people did not bother too much about that. But in 2009, the now famous genome-wide association study, it is called a large-scale genetic study, to know the how people respond to a virus called hepatitis C virus. So some people clear the virus on their own, some people don't. And some people clear the virus after taking the treatment, some people don't. So what was the reason for this? This was identified by a study called Genome-Wide Association Study in 2009. And this locus was identified as the region responsible. For example, the variants I showed you, people having one type of allele were responding better against the virus. The other group did not. So therefore, it was basically came to the limelight during hepatitis C virus genome-wide association study. In short, it is called GWAS. But since then, this locus has become important in many genetic studies. As you see here, number of diseases have been associated with these variations. For example, in pediatric children, common respiratory and gastrointestinal infections. And then in sexually transmitted infections, malaria, even cancer, and an important disease called non-alcoholic fatty liver disease that is becoming quite a bit of a problem in a country like India. It's almost reaching very large proportions affecting public health of Indians. So this is also associated with the interferon lambda 4 variant that I just explained. Not only that, as you know, are very familiar with the disease that was very prevalent of some time ago, the COVID-19, that is also associated with interferon lambda 4. But in this, it is shown that it is only in females. I will come to this aspect of the gender dependence of this association in a while. So in summary, it is very important in a lot of diseases, not only diseases as this study is shown here, in health also. For example, these are healthy women. NPC means non-pregnant, Postpartum means women who have just given birth. So if you go and measure the genes called interferon stimulated genes, ISG, example, IFIT1, IFIT2, IFIT3, OAS1, these are all different interferon stimulated genes. That means when an interferon is produced, it will increase the expression of these genes. So in these women, if you measure the expression of these genes and see the different polymorphism, different allele they have, they actually show differences. For example, the CC allele carrying women who have just given birth produce less ISG, which is basically an antiviral response. That means these women have less antiviral response compared to the other group, which have the TT allele or the CT allele, right? So a biallelic variant can have 3 genotypes. A woman can be CC, or heterozygote called CT, or minor homozygote called the TT. So 3 genotypes you can have. So in this, they have combined the one homozygote with the heterozygote, and the other one is compared to this group. So if you see there is significantly different level of antiviral genes that are expressed. So that means in health also, this is no disease, there is no virus here. In these women, they did not have any virus. But just by giving birth, they are showing different levels of antiviral response genes in them, which says that these polymorphisms are also important in health. Not only health, they have something to do with hormones. For example, female hormones immediately after birth are changing. So that has a relation, because non-pregnant women, they don't show any difference. As you see here, NPC, right? So in health as well as disease, these variants play important roles in controlling antiviral responses. That is the summary so far. Now, I told you 2 variants have been identified that control this gene, expression of this gene called interferon lambda 3. And one that controls whether interferon lambda 4, new interferon, is expressed or not. Now, since they are so close to each other, so this gene is actually present in the promoter of this gene. So they are very close physically. So, and the phenomenon called as linkage disequilibrium. Linkage disequilibrium is a phenomenon by which chunks of chromosomes get transmitted in successive generations without getting crossing over. So you know that when meiosis happens, when the gametes are formed, the chromosome that came from the father and the mother, they kind of exchange regions in between them, and that is called as recombination, right? So, but when you have this linkage disequilibrium, one of the reasons being their physical proximity, there is less chance that the region between this and this gets disrupted by recombination. For example, if you got this region from mother, you will transmit the same region to your progeny without exchanging with the region, similar region in the father's chromosome. So that is called as linkage disequilibrium. Because of that, in other words, what I mean to say is, if there is change of allele here, you will expect change of allele here also, right? Because of the phenomenon of linkage disequilibrium and physical proximity. So when that is the situation, when you associate a particular phenotype, for example, here in this case, you have taken a SNP, let us say this one, and say that, okay, IFNL3 could be the gene that is responsible for this increase in antiviral gene expression. But when this changes, this is also changed here because of this phenomenon of LD. So you really do not know whether the phenotype that you are seeing is because of this or because of this, because you cannot use a statistical test to separate out the effect of interferon lambda 4 or interferon lambda 3. This is the problem in genetic studies when you are studying variants of this nature, which involve the problem of linkage disequilibrium. And therefore, to understand, but as I told you, interferon lambda 3 is only 29% similar to interferon lambda 4, right? That means these are very different genes. They are not similar genes. So, and this gene is expressed only in some people. So, if that is being the case, it becomes important to know whether the effect you are seeing is from this gene or this gene. And the LD prevents us to use any statistical methods to understand that in the population. So you have to have alternate methods to study this. Alternate methods in the sense that you can study these proteins for their functions and and see how they are different. So that is what I pursued in my first project here. Luckily, there were commercially available preparations of recombinant protein. That means the protein which is made in a system outside. For example, this one was a mammalian cell called CHO cell line. So lambda 3 was expressed from this, and lambda 4 was expressed from bacteria called E. coli. So these were available from companies. So we wanted to study how their functions are same or different. So in other words, we asked, we could have studied their antiviral functions, right? That means the level of antiviral genes both can express. That had been pretty much studied, and it was known that they pretty much express the same kind of antiviral genes, not much difference, even though there were only 29%, identical to each other. We asked a different question. We asked a question that can they also immunomodulate? Now, what is immunomodulate? Let me define that for you. So, immunomodulation is changing the character of an immune cell. So, the question was whether you have interferon lambda 3 or interferon lambda 4, and to interact with an immune cell, whether its characteristic will change. In this case, we are looking at MDMs, or monocyte-derived macrophage, or monocyte-derived dendritic cell. These are important immune cells. They are called innate immune cells. So we expose these cells to either interferon lambda 3 or lambda 4 and look at their different characteristics, how they have changed. Now, apart from antiviral functions, these interferons also have this immunomodulation. That means their presence can change the property of the immune cell that they are interacting with. So that was the basis for this experiment. So we get from healthy individuals what are called as CD14 cells, which are called as monocytes, which become the precursors for macrophage and dendritic cells. If you remember, macrophages and dendritic cells, important immune cells. For example, dendritic cells are very important in case of getting rid of viruses from your body. Macrophages do a lot of things, including resolving your wounds, causing inflammation, number of functions they have. So we isolated these precursor cells called CD14 monocytes and then differentiated them in the lab in petri dishes using different growth factors into MDMs or MODCs. I will use these terms MDM, MODC, or alternatively just macrophage or dendritic cells in the remaining talk. Both mean the same. So when we differentiate this in presence of lambda 3, lambda 4, and then you kind of give an activation signal using what is called as a PAM, that is pathogen associated molecular patterns. These are present and are very specific for any pathogen. For example, a virus will have a certain RNA that becomes a pathogen associated molecular pattern that can activate certain receptors in the cells. So when you activate with the PAM, and then you see how these two group of cells which got differentiated in presence of two different interferon lambdas, they behave in terms of their phenotypes by using different assays called ELISA or RNA-seq or flow cytometry or even qPCR. So this is what we did, and I will explain to you what results we obtained. First, we wanted to see whether the cells, so the monocytes that I told you are also part of what are called as peripheral blood mononuclear cells that we isolate from the blood. From this fraction, we take out the monocytes. So PBMC derived, that is what it's saying here. So first we wanted to see whether it was these cells were reactive to interferon lambda 4, because lambda 3, as I told you, or maybe I didn't tell you, lambda 3 had immunomodulation properties. That means if you add lambda 3 to macrophages or dendritic cells or even B cells, it was changing their characteristics. It was known, but nothing was known for lambda 4 when we started. So we wanted to first see if lambda 4 actually responds to, or rather the dendritic cell or macrophage respond to interferon lambda 4. This is a simple experiment where the interferons generally act through what is called the JAK-STAT pathway, and the STAT protein gets phosphorylated when the interferon is acting from the outside of the cell. So if you do a Western blot, that is see the protein has phosphorylation or not, you can see whether the interferon is functioning or not. So in this case, when you put lambda 4, you see this Western blot, the phosphorylated STAT protein coming up. That means your cells are can receive signals from interferon lambda 4. Number one. Number two, I told you interferon stimulated genes, IFIT1 I told you before. So these genes, if you measure by qPCR, they are also increased when you treat with interferon lambda 4. That means these cells are responding to our protein interferon lambda 4. Now we also did for macrophages. Macrophages can be two kinds, M1, which are more inflammatory. So for example, when you get a wound, the macrophages that are formed are more inflammatory in nature. So because in the wound you might get some infection, they should be getting rid of the infection. They are very inflammatory. So they are the M1 type. Opposite is the M2 type, which is kind of anti-inflammatory. So when the wound is when the wound is healing, you see more M2 macrophages there because the infection is gone. So you don't want any more damage to your wound tissues. Therefore, macrophages become M2 or anti-inflammatory kind. So these are of two kinds. So you could, depending on what growth factors you add, you can either make M1 or M2 macrophages in the petri dishes in the lab. So in this case, we made M1 macrophages, and when we add lambda 4, there is expression changes in number of markers here. I will not get into the details of what these markers are, but these markers are also changing, suggesting that these cells are also responding to interferon lambda 4. Now we can go ahead with our experiment to see what are the differences in these two cells that got differentiated in the presence of these two distinct interferon lambdas. So we got these CD14 monocyte donors, 4 donors basically. We purchased from a company because we needed a large number of them. Now then I'm showing an experiment where the CD14 precursors were differentiated to DCs, dendritic cells. Now we did another thing called as a mixed lymphocyte reaction. Here we put the dendritic cells that we obtained from one person, let us say for example here, when you add it to what are called as T cells from T helper cells, basically CD4 T helper cells from another person, you will know that they will react, right? Because of the differences in their what is called as the major histocompatibility complex. That is why every person cannot give their organs for transplantation because of the differences. So if you mix one person's dendritic cells, another person's T cells, they will react, and these T cells function is to generate or release what are called as cytokines. Different cytokines I named here, IL-13, IL-10, etc., etc. So then you can measure. So basically you have an assay here. So I told you we differentiate the CD14 cells to dendritic cells, and then we mixed with CD4 cells that we got from different donors, about 20 per people we got. They had differences in their age, but that is not important here. Younger adults and older adults, that is not important here. But so as we measured the different cytokines from the mixed lymphocyte reactions that was derived from dendritic cells that got from lambda 3 or lambda 4 treatment, we saw some important differences. For example, the dendritic cells that came from interferon lambda 4 differentiated conditions triggered the same CD4 cells to generate more IL-4 and IL-13. Now IL-4 and IL-13 are actually antiviral, they are not proviral. So therefore, if you see here the lambda 3 differentiated versus lambda 4 differentiated cells, the lambda 4 is making the cells to secrete more IL-4, IL-13. If you have IL-4, IL-13, it is kind of bad for clearing the virus infection. So you might be surprised here how an interferon is producing something that is bad for virus infection, which is in fact what the observation in the field is called as the paradox. So for example, I told you some people can express interferon lambda 4, but if such people get an infection like hepatitis C virus, they will not clear the virus as efficiently as the people who do not have that. So an interferon lambda 4 in this case is actually hindering the clearance of virus infection. So that is called as a paradox, which we did not understand if you do just the population study. But in this experiment, we are clearly seeing why this is true, because IL-4 and IL-13, known kind of factors that are not good for clearing virus infections are increased if you have lambda 4 in the conditions. So that is one finding. Now we will go to macrophages. So as I told you, you could differentiate into M1 or M2. I will directly go to this result here. So in both M1 and M2 MDMs, we measured the cytokines released by these cells coming either from lambda 3 blue or lambda 4 differentiated macrophages. Now, as I told you, M1 macrophages are more inflammatory, but in one of the cytokine called as IL-10, which is actually an anti-inflammatory cytokine. So in M1 conditions, typically you should have less anti-inflammatory cytokine. That is why the lambda 3 differentiated macrophages show less than the control. Control is no treatment. But as if you see interferon lambda 4, it has increased than the control. That means they have kind of opposite functions here. So interferon lambda 4, I told you in the previous slide, they produce cytokines, they help produce cytokines which are more antiviral, but you have certain advantage in this macrophage because they are producing more anti-inflammatory. So that means you should always see two sides of the equation. So you may be less protected for a virus infection, but you have less inflammation. For example, that is why you see people who carry interferon lambda 4 don't see much of a problem in the non-alcoholic fatty liver because they don't have more inflammation compared to if you don't have interferon lambda 4. This is probably the reason because these macrophages are trained to produce more IL-10 which decreases inflammation. So these kind of functional assays are telling us the important differences between these two interferons which genetically are very close to each other and therefore becomes difficult to understand their different effects on phenotypes. Now we did what is called as RNA sequencing. That means sequence all the genes that are expressed in the cells, the same M1 differentiated or M2 differentiated in presence of lambda 3 or lambda 4 here. The red is those genes that get more expression compared to no treatment. The blue genes are those genes that get less expressed compared to no treatment. The treatment is either lambda 3 or lambda 4. And these are shown as heat maps here. And these are called as volcano plots. So what immediately strikes you is that interferon lambda 3, there are more genes in this box than this box, right? If you can see. This is the M2 phenotype, M2 macrophage. This is a completely different macrophage compared to, this is the anti-inflammatory macrophage type. This is the M1 is the pro-inflammatory. As you see, there are more genes when you have lambda 3 in the M2 compared to M1. Whereas it is opposite for lambda 4. If you see, there are more genes here and very less genes in the M2. And therefore, this is again an indication that they are having different functions, not the same function. Which is much more clearer in this picture here. So the red are those genes that are affected by lambda 4. The blue are those genes that are affected by lambda 3. So in M2, you have more genes affected by lambda 3. As you see, if you do a ratio, it is about 2 times more genes in M2 than M1. But for lambda 1, it is 10 times less in M2 compared to lambda 1. So therefore, in these experiments, we clearly show that these two interferon lambdas, although actually if you do an ISG expression, they will induce the same kind of genes, almost 98-99% you see same genes, but their immunomodulation property is different. When they are present in contact with immune cells, they change the nature of the immune cells very differently. As you see here, you see different kind of phenotypes. Therefore, they may be doing different things in different diseases and health phenotypes. I just showed you the association. Now, the protein is not crystallized, and therefore we don't know the structure. Whereas lambda 3, the blue one here, the structure is known, and this is the receptors that they interact with, called as interferon lambda R1 or IL-10 R beta. This is a dimeric receptor. So if you see, there were clear differences, especially with the gray one, lambda R1. If you see the number of contact points you are having with the lambda 3 and very few contact points with the lambda 4. This structure was made by homology modeling, lambda 4, whereas lambda 3 crystal structure was available. So this is adding to the evidence that these two proteins might have very different functions to each other, and therefore their effects could be different in different diseases. So you could also do the what is called as pathway analysis by using the RNA gene expression data that you get in using different databases. Here it is called as the KEGG database, or the Kyoto Encyclopedia of Genes and Genomes, where basically you see that based on the expression changes in the genes, you can map it into different pathways of known diseases and say that, oh, this disease may be important. These kind of genes are all lining up in this pathway, and therefore this disease becomes significantly highlighted. In other words, in these conditions, these genes may be affecting these particular disease processes. For example, you see tuberculosis, another disease. But what I want to highlight here is leishmaniasis, which has shown up in number of places, as you see here. M2 MDMs, just treatment with lambda 3, you see leishmaniasis coming up. Or when you compare the genes expression changes between lambda 3 and lambda 4, also you see leishmaniasis both in M1 as well as M2 conditions. Therefore, I'll towards the end, I will talk about leishmaniasis. Now, I told you the pregnant women, if you remember, the health phenotypes also can be affected by these polymorphisms. So we wanted to clearly understand, this aspect. So we wanted to test in our own local population. So we went to a population size of about 552 individuals around Kalyani, and we recruited them for this study. There are about equally 50% females and 50% males. So we collected their blood and then we genotyped for different polymorphisms that I just talked to you about. So these are different RS numbers you are seeing, but it is the same IFNL SNPs here. And now we just ask the question, let's see if, for example, these are different tests that you get done when you go for a medical checkup, right? For monocyte percent, SGOT, which tells you about your liver inflammation, or uric acid, etc. So when we did this, checked if they associate with any of these health phenotypes, surprisingly, we did see association, that is anything P less than 0.05 is significant. So you are seeing association, but as you see, it's only in males. In the females, both groups. So that means we have made the two groups based on the genotype. Either the women here carry SCC or CT plus TT. If you remember the pregnant woman experiment I showed you, it is the same here also. So if you see women, they don't have any difference. But only in males, if you see, this group of males who have CC, they have more monocytes compared to this group. And the same here, this group has more SGOT, that means little more liver inflammation compared to this group. But not in females. If you see the females, both groups look the same. And similar here. So this is the first time we have reported that there is gender-dependent, modification of the effect of these genetic variants on different phenotypes. Two things here. One thing, variants that actually control expression of interferons, antiviral molecules called interferons, they are actually influencing number of phenotypes kind of unrelated. For example, monocytes, you might think, may not have much to do. SGOT or uric acid has less to do with any virus infection, but you see association. This is called as pleiotropy. Pleiotropy, that is one thing affecting many things. And more importantly, what we have discovered is that it is gender dependent. So in males only we see this effect. Somehow females are able to counter that effect, and it doesn't matter if you have CC or the CT or the TT, your phenotypes are same here. But if you're male, that does make a difference. So this becomes important to keep in mind when we look at diseases that males get and females get, how these polymorphisms may be playing a role in terms of influencing infection and inflammation. Coming to the leishmaniasis that I was talking to you about. So leishmaniasis, as you know, is a very common problem in India, especially in the states of Bihar and West Bengal. And this is a protozoan parasitic infection. It has complex lifestyle involving a vertebrate host, humans, and the invertebrate, there is a sandfly. So the sandfly can inject what are called as promastigotes into your blood, and they go and they get phagocytosed by macrophages and also neutrophils, and they usually get settled in organs like liver or spleen, and such a condition is called as visceral leishmaniasis. And as you see, it becomes hepatosplenomegaly, enlarged abdomen. If left untreated, the patient will die, but there is luckily there is treatment, and many people will recover, but some percentage of people will go on to have what is called as post-Kala-Azar dermal leishmaniasis here. And yeah, this is about the disease. Now we were interested, there were some studies which reported like interferons. You might wonder, interferons are antiviral, what they may have to do with the parasite disease. But you know, biology is so connected and so intricate, because both are intracellular pathogens. Virus always survives inside the cell. This is also a parasite that lives inside the cell most of the time. Therefore, the immune mechanisms could be the same. So that is why we thought that some studies, as I told you, type 1 interferons they had reported, but no studies were done whether type 3 interferons have any association or influence in leishmania. So we were interested to comprehensively look at this aspect. So firstly, we want to see whether they interact. Interaction, for example, if you infect the cells in the laboratory, we can have cells, macrophage kind of cells, and we put the parasite. If there is any interaction, there should be a change in the expression level of, for example, we are measuring interferon lambda 3 here. We see that when you put interferon lambda 3 when we infect with leishmania, you see increase in interferon lambda 3 in 2 different cell lines, macrophage like cells, and then it is time dependent. You see increase as the parasite infection is going on. You see increase in interferon lambda 3, not only in human cells, and a cell line from mice called as RAW, that is also increased, both the mRNA level as well as the protein level. This was measured with ELISA. That means leishmania can induce interferon lambda 3. Now, why a parasite by inducing an interferon which is antiviral is the interesting question here. So we also wanted to establish an assay to measure the parasite load, for example. The conventional method is to do what is called as a Giemsa stain of your cells, and then manually count under a microscope what is called as the LD bodies, Leishmania dominant bodies, that will be distinguished. As you can imagine, it is kind of very cumbersome and it is error prone. So we wanted more molecular, precise assay to do this. So we used microscopy, where you could use fluorescence microscopy, because the parasite also has DNA. When you stain for the host DNA, this is the host cell taking the stain. The parasite also has DNA. This is also taken. Therefore, now you can count these, red dots. And you could do 2 things here. You could count number of amastigotes. The promastigote becomes amastigote inside the cell. So in each cell, how many parasites are there, you could count, and also how many cells are infected, that also you can count. This is one assay that you could use. But we wanted a more molecular, like a PCR assay, where we standardized by using the what is called as the kDNA. kDNA is a kinetoplast DNA that is present in the parasite. So we measure the amount of this DNA to quantify the amount of parasite by normalizing it to one of the host genes called GAPDH. That means per cell, how much parasite you have can be quantified. So we made this assay in our own hands. This is the first time we are reporting, and that pretty much the results correlated with the conventional method called LD body that I told you. So using this assay, we wanted to see, firstly, now I told you the leishmania is inducing interferon lambda 3. The next question is, can lambda 3 inhibit the parasite? Just like it inhibits viruses, can it inhibit the parasite? When we checked, to our surprise, it was inhibiting. So 100 nanogram or 1000 nanograms, if you put in the cell culture, the parasite load that you measure came down. In both human as well as, this is a mice cell line, both mice separate interferon lambda 3 is available commercially. So if you use that, both mice cells as well as human cells, by the way, leishmania can infect both mice and humans, right? So you see that the parasite can be inhibited. So that means it can act like a drug. For example, can you inject it into patients who have leishmania, like VL, and can it inhibit the parasite growth there? That was the idea. And as you see here, it was also time dependent. If you add later on after infection is given at 0 or 6 or 8 or 16 or 20 hours, the inhibition level decreases. That means if you add at early time point, only the effect you can see, both in mice as well as human cells. Now I told you interferon lambda 4, which is only 29% similar. When we tested that is also inhibiting the parasite in the cell lines. So and also we saw that not only in this case we added the interferon along with the parasite, but we also saw that if you just pre-treat the cells, that means treat the cells and then remove the interferon and then put the parasite, then also they were inhibiting the parasite load. So that means that our idea was to see if we can use this as a drug. So and how this was inhibition was being affected, we saw that the interferon lambda 3 was increasing what is called as the reactive oxygen species, which is very bad for the parasite. If this ROS increases in cells, the parasite finds it difficult to survive. So when we tested in using fluorescence assay here, as you put more lambda 3, you saw more ROS production happening, and therefore that is the reason why the parasite was not able to survive as efficiently when you don't have the treatment given. So this is one aspect of it. The other thing what we saw was using that the fluorescence assay that I told you, you could either measure the number of infected cells or the cells in infected cells or number of parasites per cell, because that will tell you whether the infection is affected or once the parasite goes inside and it is replicating, that is affected by your so-called drug. So we saw that the number of cells are decreased, but the number of parasite within the cell did not differ. That means once the parasite goes inside, it did not find it difficult for replication. That means the process of entry is affected. So that is called as phagocytosis, right? But surprisingly, when we kill the parasite, which means that it cannot replicate, you kill the parasite, you stain the parasite and then kill it so that it doesn't replicate, and then you ask the cells to do that through phagocytosis. We saw that phagocytosis is also not affected. By a complicated set of experiments, we found out that it is not phagocytosis, but some event immediately after phagocytosis is being affected by interferon lambda 3. So that is I'm not showing those here. Then model of clinical phase. So when we did the treatment in infected mice, as you see, the kDNA is the parasite load. In the liver, you see there is no difference after treatment, and the liver size is also not changed. But interestingly, we saw that we were thinking that it will inhibit the parasite, right? If you see here, the parasite load had increased in the mice who got the treatment compared to mice who did not get the treatment, and their spleens were also larger. So that means what we were thinking was different. We were thinking that lambda 3 could act like a drug. In here, we were seeing an opposite effect. So this is I told you, the complexity involved in biology. So what you see in cell culture may not completely be reflected in the in vivo system, which is what we saw. So that means mice were getting some benefit from interferon lambda 3. Maybe that is the reason why interferon lambda 3 is induced by the parasite. So parasite is getting some advantage here which you don't yet understand. And then if you go to the humans, in humans also we see this in the patients. You see the interferon lambda 3 is highly significantly increased in the patients compared to controls. That means this can never act like a drug, because if it is a drug, it should be low in concentration. Therefore, if you put it, then the parasite should go away. Here you see, these are the people who have parasite, but they have more lambda 3. That means lambda 3, similar to the mice experiment, is somehow helping the patients. We do not know that. And in a genetic association also, you see the Leishmania patients have more percentage or more frequency of the allele, which is known to increase expression of interferon lambda 3. So the contrary to our expectation that it could act like a drug, we see that interferon lambda 3 is actually proving to be beneficial to the parasite. Therefore, our effort probably should be to target interferon lambda 3 to see a drug-like effect instead of the other way around. So this is the conclusion we drew from this study. Anyway, so that from there I come to the end of this lecture. I hope you all liked my work and appreciate the science behind it. So if there are any questions, please let me know. I will be able to. I'll be either answer it right now or later on. Also, you could contact me through email, and I will be happy to answer your questions. So before going to the questions, I would like to acknowledge my funding agency, Welcome DBT, which I got funding for 6 years. Them and all the students who have done this work, and then my collaborators and our directors for the intramural funding, Professor Soumitra Das, whose time the Leishmania work was started, the present director, Professor Sagar Sengupta, and the previous director, Professor Partha Majumdar, and my international collaborators, and all the volunteers who donated the blood. Thank you very much.
Yeah, thank you, sir. It was really nice to hear to you. And the most important thing is it was understandable by me also, being a student of chemical science.
I'm so glad. Thank you.
Yeah, I have 2 to 3 questions to you. First of all, you showed actually the gender-dependent variations in the phenotype, which were taken for different female as well as male, say volunteers. So, did it on the basis of the age dependence, or the things were taken for a particular age group?
So we kind of had age matched in the males versus females. There were no major differences in the ages. Ages were the same. But the issue here is we are seeing the association within the female. So that means the 2 groups found within the females, if that is due to the age, is less likely, and we did test that too, is because you have made the groups based on the genotype, right? So for example, if you go and make the groups based on any other criteria that you look for example, this person has disease, this person doesn't have disease, etc., then this kind of confounding, age could be a confounder. You have to match for age. But this we did not make the group on our own. We went and genotype. Based on the genotype, we made the group. That means the genotype is already that was there in the population. We made it 2 groups. And because of what is called as random distribution of the alleles during meiosis, they cannot act as confounders, because what I'm trying to say is the genotype cannot be different in different ages. Yes. Therefore, age cannot be a problem factor here, because the groups are made based on genotypes which are randomly distributed in the population.
Yeah, definitely. And sir, you also showed some of the molecular modeling interactions of IFN lambda 3 and IFN lambda 4 with the IFN lambda receptor. So the interactions were studied on the basis of thermodynamic parameters, or what was the thing?
Yes, so that work was done by our collaborators in the US, right? So they first generated the homology model looking at the interferon lambda 3. Although this is not a great experiment, I told you this is only 29% similar.
Yes.
But that is the best we can do, because it's a very problematic protein, the lambda 4. It is very difficult to produce it in the lab and do crystal studies. So they did this homology kind of a thing, and then looked at probably using thermodynamic approaches, the standard methods that we use for testing interactions.
Yes, sir.
I don't know the exact details of those.
Okay, so I think it will not match exactly with that of the experimental ones, because if we do it thermodynamically also, I think there are many computational, say, limitations over there when we work.
Absolutely.
Yeah.
Unless you get the actual structure, you cannot do those things. Yes.
We cannot do those things, but that can be an aid to what
that is the best we could do. Yeah.
The best thing we have actually. Another thing is you told IFN lambda 3 increases the ROS production, I would say. Has any tests been done for the IFN lambda 4 regarding the ROS?
Yes, I didn't show the data, same exact. So I showed the data, it was inhibiting the parasite load, right? When you do the same assay, it is also increasing the ROS. Both interferon lambdas are inhibiting the parasite using the same mechanism.
Yes, sir. Because in the slide I found for the IFN lambda 3.
Lambda 3, yeah, I didn't show for lambda 4 because...
but it is also over there. And what
Yes.
I couldn't understand one thing, that was, it will be so nice of you to make me understand the PAMP, that is pathogen associated molecular pattern. What is it?
Yeah, so this is basically a signature of every pathogen. For example, as the name says, it's a pattern, right? So, for example, you might have heard of LPS, lipopolysaccharides.
Yes.
In bacteria, you do not see LPS in other species, other higher organisms. For example, humans, we don't have that thing called as LPS, but bacteria have. Therefore, the higher organisms have some receptors that recognize these things like LPS or even RNA. RNA structure will be different when you compare from one species to another. For example, although RNA is the same, the secondary structure will be different. For example, a virus will have an end of the RNA with a 5' triphosphate, whereas our RNA will have what is called as a 5' cap. So this is also a PAM. A pattern is different at the RNA for a pathogen compared to higher organisms, and therefore higher organisms' innate immune receptors can recognize this, and that is how an innate immune response before antibodies come in will be initiated.
Yeah, okay, sir, I got it. So
in other words, for example, you know about antibodies, right? Antibodies specifically recognizes a pathogen.
Yeah.
For example, your antibody can recognize even different strains of COVID, right? Similarly, before antibodies come, your innate immune molecules can recognize what kind of pathogen it is based on these patterns, whether it is a parasite, whether it is a bacteria, whether it is a virus, whether it is fungi. So this is what it is. Yeah.
The characterization of the pathogen, I would say, in this case.
Characterization of the pathogen. Yeah. Before the adaptive immune system, which has antibodies that can specifically recognize a particular protein of the bacteria or virus, before that kicks in, these receptors recognize these patterns and understand that, okay, we have some invader whose pattern is this kind, this kind. Because these patterns should be missing in our system. Otherwise, you cannot recognize them as foreign.
Yeah.
Yes.
Yeah, that is for the pathogen. So it was really nice to have you, sir, and it was spoken very cleanly and actually I would say very simply, because the matter is not so simple. It's a very advanced research, I understand. But for our students, we have I think students will be thinking, and we have recorded the whole lecture and we will be sending it to them. And
please send a copy to me as well.
Yes, sir, definitely. And whenever I will be getting the feedback forms completed by them, I will be also sharing it with you to let you know that what students have any questions in their minds. Maybe they will be very simple and trivial ones in the undergraduate.
Absolutely, that is the whole point of this program, right?
We are actually motivating our students to pursue life science as a career. So they may not have any advanced questions. I think our Dr. Shopun Kumar Ghosh has also raised a hand. Sir, can you please speak?
Hello, sir.
Yes.
Am I audible?
Yes. Yeah, please tell me.
So very good afternoon, sir. Actually, you have spoken very lucidly the most complicated things, I think.
Thank you.
In your lecture. So thank you very much for delivering such a nice and lucidly presentation for your presentation. I have a small question because I'm also a patient of non-alcoholic fatty liver disease.
Oh, okay. Yeah,
so,
a lot of people are these days.
So actually, interferons plays a key role in this case. So that is your non-alcoholic fatty liver disease and also NASH. That means your NLSH. Steatohepatitis. So which are the factors actually which triggers or which causes the progression from non-alcoholic fatty liver disease to NLSH, that is your non-alcoholic.
A very pertinent question also, because I told you in India this has become a very, very major problem. Very soon it will be large proportions of people will be suffering with this one. Now, the study that I showed you is not from India though, it's from abroad. that is only one aspect of the disease, because these are all what are called as complex diseases. Okay, complex disease, the word itself says that there are a number of things. It is not a simple one-to-one. It is not a linear kind of an association you can make out of anything. There are a number of factors that will join and cause the disease, right? Now, if you understand what is the problem here in non-alcoholic fatty liver, that means your liver is supposed to store some amount of fat. You usually get fat not from directly eating fat. It can come from glucose also. Your calories that you take as glucose carbohydrates, that is also converted. Extra things are converted to fat, right? So that is what, the liver can store some amount of fat. But if the input is so much, your carbs are coming so much, that fat starts to get accumulated in the liver. To an extent, the liver can tolerate, but after that, the liver, because of the innate immune mechanisms that are there in the liver that are designed to sense things like viruses, the same mechanisms actually start to almost consider the fat as foreign, because the fat is not normally should be present in above optimal percentages, right? So when that starts to become, the liver has to get rid of that. So it uses the same innate immune mechanism. That is why an interferon, is getting associated with this fat. So the liver tries to eliminate this fat. So almost you kind of think that it is almost like a virus that is, the liver is reacting as if it is a foreign thing. But because that is how it can eliminate that fat. So it starts to react. And some people react more than others. So this actually, the problem with fatty liver is not the fat itself, but the inflammation you get because of this. When it becomes steatohepatitis, then only you will start to see the problem. The liver enzymes goes up, you start getting fibrosis, cirrhosis, then cancer also, right? So until your liver inflammation is going up, it's not a problem. Fat in the liver is a normal thing. But when the liver starts to react against that fat, then the problem starts. So different people react, or their inflammation is different depending on number of things. One of these factor was this gene. For example, if we have the delta G gene, which we think at least 50% of us carry one copy of this interferon lambda 4, you kind of keep that inflammation lesser. That means your liver is not inflamed, which is okay, but in other words, your liver continues to accumulate fat, and that becomes a problem later on. But if you sense that and get your fat lesser, you will be doing okay. But so your question of what is the solution for this is to control your lifestyle. That is the best. Because if you can prevent the source of the problem, nothing like that. So, but once the source of the problem is not addressed, you start to do different things. For example, I was listening to Professor Shiv Sarin's lecture or interview on some channel. Professor Shiv Sarin is a very famous hepatologist in Delhi, and his advice was also this. So your drugs should be a last last resource. You should control your diet, do exercises. If everything fails, only then you should start treating, because that makes sense. Because in complex diseases, it takes so much time for the disease to accumulate, and it will take the same amount of effort through exercise, diet, other measures to get that phenotype back into the healthy state. So if you think that I can take a drug and reverse this effect, that probably will not work, because the complex disease is accumulated over time. So you have to give so much time to go back to that original health status. That means you have to do a sustained effort to get your liver back to health over a period of time. Drugs might help you, in an emergency to get your things under control. But in a long-term process, even Professor Shiv Sarin, who is an expert in NIFLD, opines this, that other measures are better than to go for drugs itself. So my suggestion would be the same. Modify your lifestyle and do a sustained effort. Don't expect results very quickly. Your exercise, your diet should go on for pretty long time. So because you have accumulated this over a long period of time. So it only makes logical sense that it might take that much time with sustained efforts to go back to that stage.
Yes, sir. Very well said.
Thank you. Thank you, sir. Very well said. Thank you.
Yes, sir. Yes, sir. So lifestyle is a very important factor, sir. Definitely, sir. So we can just, if there are no many questions, we can just get the questions by feedback. So we can just wind up the session. Let me give a very big thanks to Sridhar sir for getting for such a wonderful, wonderful lecture. And we are very happy with you, sir, and we will like to have you in person to our institution. We will be very happy to have you, sir. So participants, we can just wind up the session with Sridhar sir, and we will be meeting again in the next Science Setu lecture on 7th May by Dr. Priyadarshi Basu. Thank you all.
Thank you very much.
Thank you, sir. Thank you, sir.